Good afternoon, everyone. My name is Jeffrey Jue, and I am an Extension Educator with UMass Extension. Thank you for joining us today for TikTok with Tech Report webinar. This webinar is co-sponsored by UMass Extension Landscape, Nursery, and Open Forestry Program and UMass Laboratory of Medical Zoology. In today's webinar, Dr. Stephen Rich, Professor of Microbiology and Director of University of Massachusetts Laboratory of Medical Zoology, and Dr. Saravanan Thangamani, Professor of Microbiology and Immunology at Upstate Medical University, Syracuse, New York, will discuss what is currently known about tick-borne viruses, including Powassan virus. If you have any questions during the webinar, please type it in the question box on your control panel, and Dr. Rich and Dr. Thangamani will answer the questions. After the webinar, there will be a short survey. Please complete the survey before exiting the webinar. If you have trouble with sound during the webinar, please adjust your computer audio settings or join by telephone. To join by telephone, dial 1-213-929-4232 and access code 742-339-905. And I just also want to say that today's webinar is recorded, and now without further ado, here is the today's webinar.
Good morning, and thank you very much for joining us today. We're very pleased to have you on board to tell us about tick-borne viruses. And I think one of the things that our audience would like to hear to start off is the difference between viruses and the other pathogens that people might be more familiar with that are transmitted by ticks.
Hi, thanks for having me. So the basic fundamental difference between a virus and a bacteria is viruses are in a way they are non-living, and bacteria are living material in the sense that bacteria has all of its essential machinery within itself. It doesn't require other cell to survive. However, viruses, on the other hand, it needs another cell to survive. It has to get into a host cell to survive. So with outside the host cell, viruses will die. And viruses are a very simple genetic material. It's basically a DNA or RNA that is coated by a protein outside. Consider like a soccer ball, and a soccer ball is a protein, a structural protein. Inside, you have like a spindle structures. That's the genetic material, and that's the virus.
And in a practical sense, when people think about co-infections outside of Lyme, the canonical tick-borne disease, they think, well, most of the other ones are treatable by the same methodology that you might treat Lyme. So if you take antibiotics for Lyme, you might be killing other co-infections. Is that true for viruses as well?
No, the antibiotics that are used to treat Lyme or any other tick-borne agent, in fact, any other bacterial agent will never work against a virus. Because the antibiotics work in such a way that they try to attack the cell wall of the bacteria, unfortunately, that will not work against the viruses. So that's one reason why we do not have a specific treatment against viruses, particularly tick-borne viruses.
Mm-hmm. And the antiviral therapies are usually quite specific for the virus.
Exactly. So for treating viral infection, we need to go to antiviral therapy. But again, for tick-borne viruses, there are no antiviral therapy at this time.
Yeah, which is why there are antiviral therapies, but we don't yet have necessarily a working antiviral therapy for the big, the elephant in the room, the SARS-CoV-2.
Yeah, that's a perfect example, actually. So there are hundreds of viruses that are transmitted by the ticks, but not all of them they cause disease to humans or veterinary animals. So as you would see here, this is a nice review from Shi et al. In 2018. They kind of summarized all the tick-borne viruses under the family and order and the genus level, and you can see The ones that are important for human transmission or from the human health perspective are highlighted in this circle diagram. As you can see here, it's a Crimean Congo hemorrhagic fever virus, Heartland virus, and some of this bunch of flaviviruses that are circulating in the Americas, and also Colorado tick fever virus is here. So I can go to the next slide where it summarizes the information of the viruses that are of importance, where they are from, and what are the primary tick vectors. Again, I want to start with the Crimean Congo hemorrhagic fever because it's one of the most lethal tick-borne virus that is prevalent in majority of the Africa, also Asia, and parts of Europe as well. And they are primarily transmitted by Halama marginatum species and some other Exodus species as well. And the next one that I would like to mention is the Heartland virus that is present in the United States and is transmitted by the Lone Star tick, Amblyum americanum. And closely related to the Heartland virus is another virus called severe fever with thrombocytopenia syndrome virus. It was discovered in China, but now it's been identified in South Korea and Japan as well. And the tick that transmits this virus is Haemaphysalis longicornis, also called as the Asian longhorn tick. And that's the tick we see these days in the northeastern United States. And there are other viruses that circulates in India, Saudi Arabia. Where they affect about a few hundred people a year. And one of the viruses is actually transmitted by a soft tick. It's only one of the very few soft ticks that transmit viruses. And what I want to focus is basically what really happens in the United States is what are the viruses that are circulating in the United States that cause severe disease to humans. And as you would see here, Powassan virus is on the top list for me, and it's a primary tick vectors are Ixodes cookeyi and Ixodes marxii. Ixodes cookeyi is a groundhog tick, and Ixodes marxii is a squirrel tick. And it's been distributed, it's been prevalent in Canada, of course in USA, and also in the Russian parts. And in the map, you can see the states that are highlighted in the bright red are those states that we have found human cases of Powassan viruses. And also Powassan virus and the next one is a deer tick virus. They both actually present similar clinical symptoms. So in the map, I kind of put both DTV and Powassan as the same because it is impossible to distinguish at this time with the techniques we have if someone is infected with deer tick virus or Powassan virus. I mean, I will allude this at the later time in my talk. And the deer tick virus, as the name suggests, it's transmitted by Ixodes capillaris. The other one I will be highlighting is the Heartland virus, and that are highlighted in these blue tinge states where Heartland virus is highly prevalent. And Bourbon virus is highlighted here in the purple area, and this particular Kansas state has both the Bourbon virus and Heartland virus. And the other virus, Colorado tick fever virus, I believe that very few people may have heard about it. It's transmitted by Dermacentra andersoni. It is also called as Rocky Mountain fever, but we shouldn't confuse this with the Rocky Mountain spotted fever that is caused by rickettsia. But this is by a virus. And as you can see here, pretty much on the western United States carry this particular tick that could transmit the virus. And also human cases have been documented from pretty much all of these states highlighted in green. The last virus, it's more of thought that we should have in mind. As I mentioned in my earlier slide, this Haemaphysalis longicornis, also called as the Asian longhorn tick, is known to transmit a particular virus called severe fever with thrombocytopenia syndrome virus, or SFTSV, that has a case fatality rate of 30%, which means if 100 people get infected with this particular virus, 30% of the people will die, will succumb to this disease. Right now, the human cases have been documented in China, South Korea, and Japan. In the United States, this particular tick have been reintroduced into the United States. There is a high possibility, probably in the near future, that we may be seeing some cases of SFTSV as well. This is more of a speculative based on historical evidence.
Can I make a point here? I might actually make it 2 slides before. So if you go back to that previous slide, I think it's worth the viewers to understand that the invasion of these viruses requires not just the existence of the tick within a given geographic range, that also the virus has to be present. And for the virus to be present, very often that means that the reservoir host has to be present. So invasion, for example, of H. longicornis onto North America sans a competent reservoir for associated viral pathogens or bacterial pathogens means that there's yet another barrier that's in place to preventing that virus from taking hold in a geographic zone.
I think you said that very correctly. For any tick-borne virus to perpetuate in nature, it requires a reservoir. A reservoir is a mammal that can actually contain this virus in the bloodstream at a higher titer. So the tick has to feed on that particular animal when that animal has higher viremic titer. So I think I completely agree with you that the reservoir play a major role in introducing new viruses in new geographical niches.
I think that's important because media will tend to pick up on the expansion of a particular vector, but they won't necessarily pay attention to the whole biology that's involved with bringing a zoonotic transmission cycle in place.
So Powassan virus was first isolated in 1958 from the brain of a young boy who died of encephalitis in Powassan, Ontario. That's the name Powassan virus. And it's closely related to tick-borne encephalitis virus that is circulating in Europe, Russia, China, and Japan. In addition to that, in 1996, researchers from Harvard University, Sam Telford and colleagues identified a new tick-borne encephalitis-like virus from the deer ticks collected in Connecticut and Massachusetts. And later they found out that these two viruses are indeed genetically almost identical. So that's why Powassan virus and deer tick virus are kind of bundled together. As you would see here, that Powassan virus and deer tick virus share almost 90% genetic identity. Also, Powassan virus and deer tick virus, they cause similar or same clinical disease. Because of the similarity in the symptoms and also the serological response, vast majority of cases of Powassan DTV cannot be differentiated in a clinical setting. That's why when we kind of colloquially we tell everything Powassan virus. Unfortunately, in a clinical setting, we cannot differentiate into what exactly the particular patient may be infected with. But in a way, it is irrelevant from a public health point. Well, I should not say irrelevant, because they cause same disease. The clinical outcome is almost the same. So deer tick is now considered a genetic variant of the parent Powassan virus. So we have differentiated these two strains as lineage 1 and lineage 2. So lineage 1 is the prototypical Powassan virus, and lineage 2 is the deer tick virus. And these two viruses are, as I said before, they are transmitted by different ticks that we have in the Northeast. Primarily the deer tick virus, as the name suggests, deer ticks play a major role in its transmission, and everyone is very familiar with it, but I can walk through it. So when the female lays eggs, the eggs hatch into larvae. Larvae then find a white-footed mouse or a small mammal to feed on, and a few weeks after that, they will molt into a nymph. Nymph then can find onto larger mammals or even smaller rodents, and then they will become adult. Adult then feed on primarily on the white-tailed deers to lay eggs. So this cycle kind of perpetuates in nature. The one difference that I want to highlight here compared to the other tick-borne bacteria is that if this particular adult is infected with Powassan virus, when they lay thousands of eggs, these thousands of eggs will be positive for the Powassan virus, which means that a single female adult can actually give rise to several thousand larvae that could potentially carry deer tick virus. So that's a big difference between tick-borne bacteria and also tick-borne viruses. So on the other hand, the Powassan virus, the lineage 1 Powassan virus, is transmitted primarily in nature by Ixodes cookii, the groundhog tick. There has been reports that the squirrel tick could also play a role in the transmission of this virus, but I'm kind of focusing here on the cookii. The one big difference is that the larvae of the Ixodes cookii, they primarily like to feed on smaller rodents like groundhogs, skunks, and also robins. And the nymphs like to feed on skunks, robins, and white-tailed foxes. And obviously the cookii, they like to feed on larger mammals, again, like white-tailed fox and skunks. So this particular transmission in nature, or the perpetuation of this tick in the nature, is completely different than what is happening in the deer tick virus. Also, one has to go deep into the woods to get exposed to Ixodes cookii. Ixodes cookii cannot be found in an urban environment. You really have to go to a rural environment or deep into the woods to actually find this tick. They like to live in the burrows. They like to live in the burrows. So they are not more, I would say the deer ticks are more like a anthropogenic, anthrophilic, like they like to feed, they like to live in an urban environment where we are living close because we are living very close to the nature. But Ixodes cookia, it's very rare for a human to get exposed to it.
Probably worth pointing out also that both of those are transmitted by Ixodes ticks, whereas early on, my recollection on this might be incorrect, but that 1958, the seminal case in Powassan wasn't initially associated with a tick species.
The dermacenter.
And the tick-borne etiology came later, and it was associated with a non-Ixodes species for decades actually, right? Before it was connected with Ixodes cookiae.
Yeah, it's exactly right. I think in Canada, researchers have implicated this to dermacenter species and also Ixodes species. But when the case was identified in the United States, I think initially they attributed that to dermacenter andersoni tick. But then later, I think researchers, they did a lot of classical studies, laboratory experiments, and they have proven that Ixodes scapularis and Ixodes cookiae are the competent vector. But along the way, a lot of surveillance work went in to identify what are the ticks that actually transmit, and also what are the host reservoir that serves as an amplifier of this virus as well.
Yeah, and that may be a case of in the 60s and 70s pre-PCR, we just weren't able to make the same identification of viruses. That Dermacenter andersoni may have been a related agent, or because it's odd that we haven't seen Powassan crop up in Dermacenter since.
I think, yeah, I completely agree with you. Earlier in the time, because we didn't have the technologies that we have, I think that probably led to that. But I still believe that sometimes we do this as well. I can find that other people do as well, is that when a particular tick is positive for a virus, it doesn't mean that they actually can transmit the virus, because when a tick feeds on a mammal, it acquires the blood. It takes a lot of blood. So when we grind the tick, there is still some nucleic acid residues will be still in the tick. We can't 100% remove the nucleic acid material from the tick. So sometimes we do get, these are false negatives or false positives. Just because you can find a viral genetic material in a tick, it doesn't mean that it will be transmitting. And I believe the Dermacenter andersoni story was very similar as well. They found from the tick, but I think they never showed that they can efficiently transmit that to another mammal. So I want to kind of highlight the distribution of Powassan virus within the United States. As you could see here, that majority of the cases are bundled around the Northeast United States and also the Great Lakes region, with Minnesota and Wisconsin being the, where majority of the human cases have been identified, followed by Massachusetts and also New York State. And over the years, we have seen that this particular distribution has been actually moving southwards. As you can see that if I can overlay with the distribution of Ixodes scapularis ticks, the deer ticks, you can clearly see that it is actually expanding to wherever the deer ticks are actually currently distributed. So what I'm thinking is that, it's just my pure speculation, is that potentially in the next decade or so, we may be seeing Powassan virus going all the way to the Texas, because that it basically overlaps the geographical distribution of the deer ticks. Together with the geographical distribution, so one thing that in the previous slide, I was kind of alluding to the geographic expansion of the Powassan virus to different states. It originally started in the Greater Lake, New York State area, and then it kind of went downwards, southwards, basically. I want to show these statistics also to show not only the geographic expansion, also the number of human cases are on the rise. So you can see that the virus was discovered in 1958, and nearly 34 years from that, like 40 years, we only saw 0.7 cases per year. We didn't see much. But 10 years after that, we started to see 1.7 cases per year. And then from 2009 to 2015, the cases went to 8 cases. So what I'm talking is human cases, so 8 human cases per year. However, in the last 4 years, there is a steep increase in the number of human cases. we are on an average 27 cases, 27 human cases per year. So it's a clear indication that the Powassan virus human cases are slowly on the rise, and particularly in the last 4 years, there is a dramatic increase in the Powassan virus cases. It's something that I want to kind of highlight before I move on to the next slide, which is the clinical manifestation. So Powassan virus is an encephalitic virus, which means it causes encephalitis, meningoencephalitis, and aseptic meningitis. There are two phases of this disease. The first phase is called prodromal, or the acute phase infection, which is lethargy, headache, sore throat, and disorientation. The encephalitic phase, classical vomiting, prolonged fever, respiratory distress, loss of coordination, speech difficulties, and seizures. And Powassan virus is a fatal disease. It can cause fatal neuroinvasive disease in up to in 10 to 15% of the cases. Of those, 50% of the survivors will have long-term neurological sequelae, including headaches, muscle wasting disease, and memory problems. And some infection may only cause a febrile illness and may be asymptomatic. And that actually can very well discuss in this schematic diagram. So the incubation period for Powassan virus is almost 1 to 5 weeks. In the first week, one would experience a febrile illness like fever, sore throat, drowsiness, headache. It's a common febrile illness that is associated with any illnesses, basically viral illnesses. From weeks to month, if one's immune system is strong, they will recover from the febrile illness and they will be asymptomatic and they will be cleared out. They will clear the virus from their system. But if their immune system is not strong enough, then they will go into the encephalitic phase, which is fever, vomiting, respiratory distress, paralysis, and seizures. 10 to 15% of these cases, encephalitic disease patients would succumb to the disease. However, if they are lucky enough to survive, they would still, 50% of the survivors will have long-term neurological sequelae. And I have this picture just to show how severe the disease is. This is an MRI image from a fatal case of Powassan encephalitis on admission. This is on admission. And after 8 days, this is the brain, showing severe inflammation in the cerebellum, thalamus, and midbrain. And this patient lost consciousness and capacity for independent respiration 2 days after admission and died on day 13. So it's a very quick, fast-moving virus. If someone's immune system is not strong, it will actually go to this center, infect the central nervous system, go to the brain, and will inflame the brain, cause meningitis or meningoencephalitis. And I can give you an example of the clinical manifestation with North Carolina Senator Kay Hagan, who died of Powassan virus recently. So she acquired the infection in 2016. She was hospitalized with encephalitis. So from 2016 to 2019, she had this neurological sequelae. She recovered from the initial encephalitic phase, but the long-term neurologic sequelae, she succumbed to that and she died 3 years after she originally got the infection. So this is a classic case to show that even if you survive the initial encephalitic phase, your quality of life will be affected, and this could eventually lead to the death as well.
Might be good to interject here, and you may have data coming like this as well, but we test human biting ticks for presence of Powassan, and we're up to about 30 now that have tested positive for Powassan, and we do as a courtesy, we follow up with those individuals that have been bitten by those Powassan positive ticks. And to date, none of them have been symptomatic. So it's probably worth noting that we're talking about Powassan infection and not necessarily Powassan exposure.
Yeah, I think you said it very well. I think most of the cases that, in my opinion, I think I've been working with Powassan for nearly 13 years now. Mm-hmm. And I think several of the cases, they go asymptomatic, which means that, as you mentioned, that maybe they were bitten by a Powassan virus positive tick. They are exposed to the virus, but they never got sick. They may have gotten a febrile illness like any other fever, viral fever, mm-hmm. but then they might have gotten over it. So those are the lucky ones, I would say. But unfortunately, that's not the case for a lot of people. But I would say that, as we know, that over the years, at least particularly in the last few years, there is a steep increase in the human cases. So yeah, we have to be cognizant with the fact that not all Powassan infected ticks, when they feed on people, that will result in encephalitic or neurologic sequelae disease.
I just think it's sometimes valuable for people to understand that this may be more like a West Nile virus, which is a serious virus, but has limited impact on many of the people that it infects. It's more like West Nile virus than it is like Ebola virus.
You nailed the point correctly. So Powassan virus is very similar to West Nile in terms of how several people will be asymptomatic. Only a few will actually go through the encephalitic phase. And it's definitely not like an Ebola or any other hemorrhagic fever viruses that has 50% case fatality rate. So I want to kind of give this statistic as well to show what the seasonal distribution of the Powassan cases. And this kind of corresponds, so you can see from beginning of April all the way to November, December, peaking in June. And this kind of mimics what the deer tick would actually have the seasonal prevalence as well. So it's not surprising. What are the diagnostic methods that we have right now? As I mentioned earlier, the incubation period is 1 to 5 weeks. And in the first week, one would actually get very like a febrile illnesses. So at that time, viral isolation or PCR is the only method. And the viral isolation can be from your serum, the blood, or from the spinal tap, or from the cerebrospinal fluid. From weeks to months, it's primarily serological test based on the blood, or also from the cerebrospinal fluid. Unfortunately, this early period is very difficult to detect because the virus circulates in the blood only for a short period of time. Once you get infected, once a tick bites you, within the next few days, ticks kind of go through the circulatory system in the blood and go all the way to the brain. So only for a day or two, one might see virus in the circulating blood. And then it immediately infects the central nervous system and stays there. So catching the virus at this time is really impossible unless and until the clinician knows exactly what he or she is doing. Most often, cerebrospinal fluid is the one that it's been taken and sent to reference laboratories to actually identify the virus.
Are people likely to seroconvert despite the low viremia, even absent onset of encephalitis?
Yes, they would have a low level seroconversion, but obviously the rate is depending on the dosage of the virus that they got, but they would have seroconversion. With that background, I want to actually touch one topic about the perpetuation of tick-borne viruses in nature. Is that you can see from the picture on this left panel here, it actually nicely illustrates the life cycle of a tick and how each life cycle of the tick would actually feed on smaller mammals, like larvae would actually feed on smaller mammals like mouse primarily. This is meant for Ixodes ticks. And then the nymph, they like to feed on a little medium to large sized mammals like raccoons, spotted skunk, gray fox, woodchucks, skunks. And the larger adult will like to primarily would feed on larger mammals. I think deer or elk or moose for the Ixodes species that are in North America. And for the Ixodes species in Asia and Africa, it's primarily goat and sheep. So I want to kind of give a concept here or try to explain a concept here as how viruses are perpetuating in nature. So for a virus to successfully survive in the nature, continue to survive and infect humans or animals is that they need three components. They need the virus to be competent to infect, they need a competent tick, and they need a competent reservoir. So what do I mean a reservoir? Reservoir is an animal that becomes infected with the virus and keeps the virus circulating in his bloodstream as long as possible and in high titers enough to infect a feeding tick without dying from infection. So this animal has to survive the infection and the tick has to feed on it and the tick should acquire it. So you need three components to coexist together in such a way that you have to have this coexistence in such a way the virus can perpetuate in nature. There are conditions where ticks by themselves can act as a reservoir. What I mean that is that they can also transmit the virus to other ticks without the need of a viremic animal, without the need of an animal reservoir. What I mean is that I can illustrate that with this small schematic diagram. A reservoir host, normally what I mean is an infected tick feed on non-exposed, let's say mice, for example here. and then it delivers a virus, and the virus replicates in the reservoir mice. The next time when a naive tick, which means the tick that never had virus before, feeds on this viremic mice, it then acquires the virus. All the dotted things are viruses here, things that are looking the Y are antibodies against this virus. So they get this virus. The fourth situation is where, so when this virus, when this particular mice or animal reservoir is exposed to a lot of virus, naturally, its immune system will develop antibodies against this virus. So at this stage, the animal will have a lot of antibodies. So when a tick tries to feed on it, the virus acquisition will be blocked because of this antibody, because there are no virus in its system. It's all clear. So that's called the natural reservoir host. That's how all the natural transmission happens for ticks and mosquitoes and everything. But when I say that ticks can act by themselves as a co-feeding reservoir is by the mechanism called co-feeding. That's where when multiple ticks feed on the same animal, like in this case, the first one, number 5, it's called co-feeding, that enables the tick to pass through among themselves without the need of viremic house. So what happens here is you can see that this is a viremic tick. This tick, let's assume this is positive of Powassan virus. This is a naive tick that does not have any virus in it, but they are feeding so close to each other. So what happens is that whatever virus that is injected by this particular tick is taken up by the naive tick that is feeding very close to each other. So basically it eliminates the need of having a viremic host. So it does not need to have this particular viremic host. Just a naive animal is enough. So it circumvents the need to have a viremic host. That's what we call it co-feeding. And this co-feeding can happen even if the mice has antibodies against Powassan virus. So that is the interesting phenomenon, how ticks can act as a reservoir or amplifier host for its own virus that is transmitting.
And some viruses may be transmitted exclusively by this means, right? For example, Crimean Congo hemorrhagic fever may be a viral infection of ticks.
So the prevailing theory is that co-feeding transmission is the primary way how tick-borne viruses are perpetuated in nature.
All viruses, all tick-borne.
That's the prevailing theory, yes. That's a prevailing theory, but I think several studies have been done by scientists in Europe to demonstrate that. And I think we are slowly starting to see that phenomenon for Powassan virus as well. Reservoir host, this 1, 2, 3, this particular thing will happen, but this is probably what we are assuming is that what we are actually seeing in our data and also previously published data is that this is what is happening in nature. That's how the ticks transmit each other the virus and survive in the nature.
Does the virus integrate in the mammalian cells? Can it express protein from within mammal cells?
Yes, it actually can replicate. Tick-borne viruses can replicate both in mammal cells and also tick cells. I don't think there is a problem with that. So treatment and prevention. There are no FDA-approved vaccine or specific therapy for Powassan virus. And for Powassan cases requiring hospitalization, supportive care is provided and it often involves respiratory support, intravenous fluids, reducing cerebral edema. Minimizing tick exposure is the best preventive measure for Powassan virus infection and also other tick-borne agent infection. However, there are vaccines available for tick-borne encephalitis virus. That Powassan virus is very close to the tick-borne encephalitis virus. If you go to Europe, you can get this vaccination, and this particular vaccine can protect with 50 to 60% efficacy, basically. So just an informational thing is that anyone who comes to my lab who wants to work with Powassan virus, I normally send them to Germany to get this vaccine. This is actually, it's a Baxter vaccine. They go to Germany, go to one of our collaborators' lab, and he's an MD doctor. They go to his lab and they get a vaccination. So we need to have 3 doses. So there are options. You can go to Europe and get it, but it's not 100%. It doesn't protect 100%. Experiments have shown that it only gives 60% cross neutralization. So
60% in protection against Powassan or against TBE?
Against Powassan. So this particular vaccine, it's more like it can cross-neutralize a lot of tick-borne flaviviruses because these are all in the flavivirus family.
What about West Nile? Would it protect against West Nile?
Not really,
No.
because they use different protein, different entry mechanisms. So it's a little bit different.
Mm-hmm.
But it's a good thing we can actually try to understand the cross neutralization. But that's one reason that these viruses are very different. The only reason why the tick-borne encephalitis vaccine works against the Powassan infection is that they're so closely related and they're genetically identical to 80 to 85%. So that's really, really high homology. So that's one reason why. And also they use a similar infection mechanism, like the way that how they actually is delivered in the skin and then goes all the way to the central nervous system and go to the brain. Tick-borne encephalitis uses the same way as the Powassan virus. So the mechanism of infection and the tropism, they're almost identical, and that's one reason why we get almost 50 to 60% protection.
How does FDA feel about an off-label use of a vaccine? And are there efforts to make this an FDA-approved protection against Powassan?
I have asked this question multiple times. Why, when a vaccine is approved in Europe to be given to humans, and it's been there for like 15, 20 years, why we are not doing? I honestly, I don't know the politics behind it. I know for sure, like countries like Austria, almost everybody gets a vaccine there, and the tick-borne encephalitis cases are on the on the negative. Almost it's going down there because everybody's getting a vaccine. You can go to a pharmacy store and get a vaccine there and get vaccinated, and that's something that I can't understand why we don't. But you can go to, I think 2-3 years back or prior to that, you could actually go to Canada, go to a travel clinic and get this same vaccine. But now it's been taken off because I think that it's not enough commercial interest for the companies to actually, put this into the market. But the other thing I should have mentioned is that my lab used to work with tick-borne encephalitis and the BSL-4 before. And that was another reason why all of my lab members went to Germany to get it, because that is one of the requirement for my earlier institution to have this vaccine if someone wants to work with tick-borne encephalitis. So that was one of the reasons why we really went there.
Mm-hmm.
So with that, I want to kind of, move on to other tick-borne viruses that are recently discovered and are potentially dangerous as well. The first one I want to talk about is the Heartland virus. Heartland virus was first discovered as a human illness in 2009 in Missouri, and colleagues from CDC, Macmillan, and others, they discovered this particular virus. And later, CDC entomologists identified that Lone Star ticks are the vectors that carry this particular Heartland virus to infect people. And as of September 2018, there are about 40 cases of Heartland virus disease, and primarily in the Midwestern and Southern United States, as you can see from the map here. And most people diagnosed with this disease became sick during May through September. Kind of coincides with the tick seasonal distribution as well. Right now, we do not have any treatment or vaccine against Heartland virus. It's only by supportive therapy. And antibiotics, as I said earlier, doesn't treat viruses. So the interesting phenomenon I want to highlight here is that, so as I said earlier, we have about, in the Midwestern and Southern United States, the case have been identified. However, there was a study that was conducted by CDC scientists, and they published in 2015 in Emerging Infectious Diseases. What they did is they actually tested the seroprevalence of Heartland virus antibodies in the wildlife population that was collected from the southeastern United States and also the eastern seaboard of the United States and the southern United States. As you would see clearly here, the states that are highlighted in red, they had seropositive wildlife. That is in 2009. So this serum was collected in 2009. So basically, when the human cases were being identified here, wildlife in this particular state, they were already having antibodies against this particular virus. So that's an interesting phenomenon, which which makes me to believe that this virus had been circulating in nature for a long time. It never crossed, we never actually, identified a human case, and 2009 we identified a human case. So that's an interesting phenomenon that I was pretty excited to actually see that.
Tick reports detected the Heartland virus on Long Island now, where Lone Star ticks are pretty much overcoming.
Oh, Long Island is, I have a feeling that I may be wrong. You can tell me correctly, as you have more ticks coming to your tick testing center. What I see from the published literature and also what we are getting, we get more Lone Star ticks from Long Island area compared to Ixodes. So it makes me to believe that Ixodes, the deer tick population, is slowly being replaced by Emblema americanum. Do you think is this happening?
Yeah, we're seeing the same thing, and it's a curious phenomenon, because one would have to wonder how they're competitively excluding the deer ticks. Like the Lone Star ticks, they breed and they live all three stages on white-tailed deer. So it could be they're just getting a better bite on the white-tailed deer than the black-legged ticks, which is curious.
Yeah, I think this is something that one of the graduate students in the lab was kind of thinking about it. Okay, why, how is this population replacement happening? How come this Lone Star ticks are trying to take over, and then somehow the Ixodes deer ticks are kind of disappearing from there? Also, I think I believe that the Asian longhorn tick is also kind of appearing in that area. Mm-hmm. So I'm not sure how this interaction of these three tick species that are competing for resources in a similar, in a single geographical environment, mm-hmm. and consequences of that, that would be interesting research to look into that.
Could be good for Lyme and bad for other things.
That's a good point. Yeah, you will see less Lyme cases, but the only thing is that, as I pointed, the seroprevalence of this Heartland virus is all the way up to the Canadian border, which means that they didn't get any samples from New York or anywhere from the Long Island. But it makes me to believe that the wildlife in the Northeast probably had been exposed to the Heartland virus sometime in the past. Also, Lone Star ticks are pretty aggressive feeders, and they can chase us down. That's why we call it hunter ticks. Unlike the deer ticks, we have to go to the deer ticks, but the Lone Star ticks can actually come to us and feed on us. Also, they're very aggressive feeders. So I think that we can only speculate that the Lyme may go down, but the other tick-borne diseases will probably crop up in the horizon. So the next virus of interest that I want to talk is the Bourbon virus. As the name suggested, it was identified from a man in Bourbon County in 2014. That's why it's called Bourbon. It was discovered in the state of Kansas, and he died after getting the tick bites. And however, I think the death was attributed to comorbidities as well, but it's not 100% clear. But I think we need more research onto this virus to really understand the significance of the pathogenesis and what really happens to a human upon getting this particular virus. So patients with the Bourbon virus normally report fever, fatigue, anorexia, and nausea, vomiting, and maculopapular rash. And they may also see thrombocytopenia and leukopenia. And some of the patients found to be infected with Bourbon died during the acute phase illness, but we still don't have an accurate number, how many people are being infected with this thing. So it's something that we need more scientists to come into this Bourbon virus research to understand more what is happening. Also, this is a potential, this is transmitted amblyomma that is slowly making its way in the Northeast. So another virus to actually look out for in the horizon. And the Colorado tick fever, this is surprising to me. That, honestly, I didn't pay too much attention to this, where I know this virus is there, it's circulating in the western part of the United States, but the more and more I started to read about this virus, it kind of intrigues me. So this Colorado tick fever was initially recognized in the early 1850s as one of the various Rocky Mountain fevers. And so I should mention again here that this is nothing to do with the Rocky Mountain spotted fever that is transmitted by rickettsial pathogens, rickettsial agents. And ironically, or coincidentally, it's the same tick that actually transmits both that rickettsia and this virus, Dermacentra andersoni. It's a Rocky Mountain wood tick. And in 1940s, Topping and colleagues actually started to call this Colorado tick fever. they showed some interest in identifying and isolating this agent, and eventually in 1940 they were able to show that. And in 1950s, Flory and colleagues demonstrated and also isolated this Colorado tick fever virus from Dermacentra andersoni in nature, and also they have demonstrated in the laboratory. And so far, from 2002 to 2012, there have been like 83 human cases reported from primarily the western part of the United States, and the counties that human cases were reported are in the darker blue compared to the pale blue. The pale blue is where the Dermacentra andersoni has been known to be present, and the dark blue are where the human cases have been documented. So the incubation period for this particular virus fever is 1 to 14 days. The most common symptoms are fever, chills, headache, body aches, and you will feel tired all the time. And about half of the patients will have biphasic fever, which means that you will have several days of fever, feel better for several days, and then you will have a second period of fever and illness again. And most people who became ill have mild disease and recover completely. However, weakness and fatigue may last several weeks. In rare cases, some patient may develop severe illness that affects the central nervous system with symptoms that include stiff neck and confusion. Life-threatening illnesses or death due to the Colorado tick fever virus are very rare. It makes you sick, but it doesn't necessarily, you will not succumb to the disease. So I like this picture that I took it from CDC that actually tells the ecology of tick-borne virus. This particular virus is not will not transmit from adult to larvae. So basically, when the larvae, they primarily feed on squirrels and chipmunks, and mice. And if these the animals usually do not become ill, and they pass on to the virus to the other ticks, like an animal reservoir. And when the nymphs then feed on the smaller rodents as well, and adults, they try to feed on larger animals like elk and marmots. Also, humans become accidental hosts for this tick when we do hiking and other things. So, and also the other thing to note is this particular tick lives mostly in United States and Western United States and Canada, primarily at 4,000 to 10,000 feet above the sea level. So and the human cases kind of correlate to this particular distribution of the tick as well. The virus is not transmitted from person to person, except in rare instances of blood transfusion. The reason being is that this particular virus can remain in the blood cells for several months, and in blood and bone marrow for 6 months following infection. So that's why I think it is advisable that if someone is tested positive for Colorado tick fever virus, it is advised not to give blood donation for at least 6 months. So with that brief introduction about the ticks viruses that are circulating in the United States of importance, I want to touch a concept of the timeline of tick-borne pathogen transmission. As everyone knows that tick-borne pathogens are not transmitted at the same time. Every pathogen has its own time when they are actually delivered to the feeding site. Here I can use an example of Powassan virus and tick-borne encephalitis. They are closely related. Our lab and other labs have demonstrated that Powassan virus can be delivered as soon as the tick latches onto the human body or a mouse body. It has been demonstrated for tick-borne encephalitis virus as well, and I can show you an example here. And this is the feeding mouth part where the tick is kind of latching itself, and you can see the positive cells that are highlighted in the pink. They are positive with the virus, and this is right at the feeding site. So you can appreciate that the virus is dumped. This is as early as 3 hours. This image was taken 3 hours. And it was the same for Powassan virus as well. So although it's not as good as this one, but you can see here, this is actually the feeding mouthpart. The tick cement is here. And these are the Powassan viruses, the ones you see in the pink. Those are Powassan virus infected cells that are right underneath the feeding site. As soon as these are at the very early stages of the tick. So that tells that, that's why we conclude that tick-borne viruses are actually transmitted to the feeding site as early as ticks start to feed. And from the literature, Anaplasma rickettsia and also Borrelia miyamotoi, they need anywhere from 12 hours to 24 hours to actually to be deposited at the skin site. And Borrelia, from the literature, it's anywhere from 24 to 48 hours. And that's skimming through the literature. I know that there are some odd research articles that tells a little bit earlier, but on an average, this is what the general understanding of the researcher is. Borrelia, or the Lyme disease agent, requires at least 24 hours to be delivered to the skin site. And I will tell you with a very cartoonish reason, a cartoonish picture to show why this is actually happening, is that the top panel is for Lyme disease agent transmission. So this is a tick feeding site. This is actually a cross section of a feeding site. This is the tick body. It inserts its mouthparts deep into the subcutaneous space. And this is a mouse skin. This is from one of our research study. I can say that, so when this, let's consider this is an infected tick. So in an infected tick, the Borrelia, the red color spirally ones, let's assume it's Borrelia. So the Borrelia stays in the midgut. It doesn't go anywhere else. It just stays in the midgut. But as soon as the ticks start to feed, as soon as the blood from the mammal goes to the gut, it sends some signals to the Borrelia in such a way Borrelia gets activated. So when I say activation, means that it will start to reproduce, it will start to multiply, and then from the midgut, it will move out of the midgut to the hemocoel, to the circulatory environment, and then go to the salivary gland. From midgut, it goes to salivary gland. So activation upon blood feeding, and then the Borrelia replicates, multiplies inside the midgut, and start to escape the midgut, slowly move to the salivary gland. Once it goes salivary gland, it multiplies again, and then it gets dumped into the feeding site lesion. So we get infected. So this entire process of activation to the transmission takes at least 24 hours. And that's why Borrelia infected tick, we normally say that we need 24 hours to 48 hours to get transmission. So, however, on the other hand, for the viruses, it's completely different. In an infected tick, they will be in the salivary gland, they will be in the midgut. In fact, the entire tick will be infected with the virus. So there is no activation, there is no migration. It's already there. So when an infected tick latches on to a human or an animal, it dumps the virus right away. And that's one reason why I believe, I'm an advocate, I think based on the literature, that's what I believe, is that the viruses are able to transmit right away. However, particularly the Lyme disease bacteria, it requires at least 24 hours to be transmitted. And this is just want to kind of highlight the same thing what I just said, that basically there's a reason why Borrelia takes a little longer to be delivered to the skin site compared to the viruses. And I want to finish my talk with this particular slide that I love, because it was one of the, it was a picture taken, it was a study, thesis study done by one of my first graduate students and was published. This actually compares the tick feeding site of Ixodes capillaris and Dermacenus andersoni. So we all know that Ixodes capillaris transmits Powassan virus, and Dermacenus andersoni, as I just alluded, it transmits Colorado tick fever virus. And just look at the feeding, how actually, this is an nymphal tick. It actually put this mouthparts deep into the subcutaneous space, and it dumps a cement around the feeding, at the laceration site in such a way that it glues the tick so that it can continue to feed. And it goes deeper. However, on the Dermacenus andersoni, look at this. It actually doesn't even penetrate the skin. It stays in the subepidermal layer. It barely penetrates. And it dumps a lot of cement protein, like a glue protein, so that it can keep the tick attached and start to take the blood. And this is 12 hours post-attachment. This is 48 hours post-attachment. You can see that the amount of cement that is deposited and is barely touching the epidermal layer. So why I'm talking about this is that no two tick species are the same, which means that what we know about Powassan virus that is transmitted Ixodes capillaris will not be applicable to the Colorado tick fever virus that is transmitted, because they all use different mechanism to transmit. They all use different mechanism to activate the skin cell response in such a way that they are taken to the organ of interest and cause disease. So I just want to highlight that no two tick species are the same, also no two pathogens are the same.